And just want to acknowledge that I'm speaking from the area of the Coast Salish people, the unceded territories of the Musqueam, Squamish, and Tsleil-Waututh. So we're very pleased, at least I am in particular pleased, to talk about something other than COVID for a change, and to share with you some of the work we've been doing over many years and formed the basis for Liz's PhD, is to talk about how we get to cervical cancer elimination in women living with HIV and some of the work we're doing to try and contribute to that. Just in terms of disclosures, we have been supported by Merck for some of this work with in-kind support for vaccines and assays, and funded by CIHR and the Canadian Trials Network, but also I've had some funding from other pharmaceuticals for vaccine trials, and Liz is funded from Michael Smith and CTN. So we're just going to do a little bit of background and then talk about some of the early HPV and HIV studies, and then I'll hand over to Liz to talk about some of our somewhat more recent and our plans for future studies. So I think many of you may be aware, and forgive me for reviewing sort of basics for a gynae cancer group, but specifically talking about cervical cancer, although I think many people in the public think it's a long gone disease, it's still common globally, 500,000 cases annually and 300,000 deaths. And we still have significant number of cases in Canada with 400 deaths, so it's not a trivial problem. But relevant to what we're talking about today with regards to HIV co-infection, cervical cancer burden is highest in many low and middle income countries, and sub-Saharan Africa has the highest rates and high mortality associated with it. I think many of you are more than well aware that this is one of the cancers that is actually caused by a viral infection. Human papillomavirus actually is the cause in almost all cases of cervix cancer, and in many cases of other genital cancers as well as head and neck cancers. It is the most commonly sexually transmitted infection, and many people are exposed and acquire it, although fortunately briefly, and clear it in many cases. These viruses are divided based on their behavior into high risk of oncogenic transformation versus low risk. And 16 and 18 certainly have been the most common, but not so much in those living with HIV, which Liz will share later. The issue with regards to HIV infection is that it's pretty clear that, as I said before, many people are exposed to HPV, acquire it briefly and clear it, but you require a really robust, primarily cell-mediated immune response to facilitate that clearance. And women living with HIV have significantly higher rates of HPV infection and significantly higher rates of cervical dysplasia and cancers. And it appears that it's a combination of the immune dysfunction and some actual facilitation by the HIV virus itself that increases the oncogenic potential of HPV in women living with HIV. Now, I will just mention that some of the work we're presenting today on the vaccine is actually a multi-decade Canadian collaborative effort where Kate Hankins led on looking at these rates of HPV infection and cervical cancer. And we did see, interestingly, a reduction in the impact by effective antiretroviral therapy, which boosts the immune response and the immune function in people living with HIV. But it's important to realize that people living with HIV represent a huge number of individuals globally, so nearly 20 million, and still almost 15,000 in Canada. So this is a population of interest and importance for HPV and cervical cancer. There's also, as I mentioned, some data to suggest that HIV itself actually enhances HPV's ability to have early gene expression, which is part of the pathway for cell change that results in intraepithelial lesions, precancers. There's also a diminution in the ability of the immune system because there are fewer antigen presenting cells in persons living with HIV, so a difficulty in clearance of the HPV, and then less circulating cytokines that also are effective in clearing HPV. So I think many of you may be aware that the WHO called for cervical cancer elimination, which is actually quite an extraordinary thing. The WHO actually doesn't call for elimination of that many diseases, and polio being the most, one of the most notable. But in 2018, this was felt to be an achievable goal for the world, and that the idea was it would go to less than 4 cases per 100,000 women years. So this requires vaccination of youth, a high degree of screening, and of course, and then a high degree of treatment. And all of those things have different challenges in different parts of the world and in different populations. And so there is a life course approach, but I think it's important when we look at this life course approach to realize that when persons have HIV, this gets shrunk a bit, and the timeline to go to precancer and cancer actually gets tightened and shortened. So it means vaccination is even more important, and screening and management also extremely important. Again, not new news, I'm sure, but there were originally, in the early phases of HPV vaccine development, a bivalent vaccine with just 16 and 18, so really targeting cancer, whereas there was also a quadrivalent vaccine that also added 6 and 11 that is responsible primarily for the benign disease such as genital warts and respiratory papillomatosis in children. And at the time when we initially planned studies that we're going to talk about in a sec, the only licensed vaccine that just at that moment in time was the quadrivalent vaccine. And with all due respect, the company Merck that produced it was really willing to engage in an investigator-driven trial. So there's now the primary vaccine in our country is the nonavalent vaccine with many more subtypes covered. And we have great safety data, high immunogenicity, high efficacy in general populations. And much of this data comes from British Columbia. But background of knowing that there was a reduced response in people living with HIV. These maps are here just to superimpose HIV and cervical cancer globally. And I don't know if you can see my pointer, but the dark colors, the sort of more red dark colors are higher incidence, pale being lower. And that here you see a huge burden in sub-Saharan Africa of cervical cancer incidence and mortality, and to some degree in South America. And then we see vaccine programs not being present in many parts of these vulnerable countries. And this is the prevalence of HIV in the world by color depiction, where many countries in Africa have 1/4 to 1/3 of their population of young women with HIV. So it's a perfect storm, unfortunately, for HIV facilitating HPV infection and cervical cancer, and programs that prevent it not being present in these low and middle income countries. So, so that essentially is the rationale of why we need to understand the HPV and the HPV vaccine in this population. And this is really of utmost importance. And if we're going to eliminate cervical cancer globally, we can't ignore the interface of HPV and HIV. So just a little bit of the early findings, and then I'm going to hand over to Liz in a sec. So right at the beginning, and this is going back a while now, hard to believe, but we started in 2008 with a CIHR funded phase 1 study with the HPV quadrivalent vaccine at the time. And the idea was we wanted to determine what the seroresponsiveness was. We didn't know if it was going to be the same kind of response in these immunocompromised individuals with a co-infection. And then we wanted to at least have a look at the effectiveness and look at the response to the specific HPV genotypes. And we, again, historically had some data to suggest persons living with HIV had higher rates of adverse events, and we wanted to see if that was the case here. So this has been an extraordinary collaboration, which has pretty much continued on to this day, with this huge number of individuals across the country and their teams willingly participating with a modestly funded CIHR funding source. This is the overall mapping. So there's a pre-screening. I don't know why it's doing this. This is so weird. Pre-screening, then the vaccine, then data collection, and then and it was a 026 licensure planned sequence of vaccines. We also had great collaboration with we wanted to use the Merck CLIA assay because that was where all of the data globally at the time was that same assay. So we wanted assay to assay comparability. The Coutelet lab in Montreal, François Coutelet did our HPV DNA testing on his linear array. And we had the BC CDC, obviously, or BC Cancer Agency, I should say, reference lab, do the cervical cytology, and then clinical sites primarily for the other measures. We were able to enroll 351 girls and women. And for those of you who don't work in HIV clinics and realize that the population of persons that attend these clinics are often poor, marginalized, immigrant, higher rates of rural indigenous persons, and sometimes quite difficult to engage in care. And we were able to get them to engage in this study, mostly because they really, really wanted to be. So we had both high engagement and high completion of 3 full doses. The good news is that's good for them. The bad news is it left us with not so many numbers to look at subset analysis of 1 and 2 doses. But we were very pleased with this outcome. A lot of work on the part of our clinical sites. And this gives you a sense. We included all ages because of the demographics of our clinics. We had a modest number of young adolescent girls. We do have 10% identify as Indigenous and almost half Black, because this is a reflection of the clinic's populations, not of Canadian, but useful to us for international comparisons. Many were well engaged in care, but only 70% with a full effective antiretroviral treatment and undetectable viral load, which is the goal for those of you who aren't involved in HIV. And here was an extraordinary seroconversion rate at 1 month after the 3rd dose, with very high rates, much higher than we actually anticipated, with the exception of HPV 18, which is always problematic. And just a little bit of information on this. So having a suppressed viral load is in the kind of bluey color, and I guess, I don't know, maybe turquoise for unsuppressed, and you can see significantly different seroresponsiveness based on whether or not they were immune reconstituted and virally suppressed with antiretroviral therapy versus not. So felt very much here that the message is, if you have the opportunity, get people successfully treated with HIV before vaccination. And then what we did is we took our HIV population and compared them to published data of HIV negative individuals in a comparable age group. And we're able to show that at the early phase, persons who were HIV negative had higher immune responses, but this blunted by month 24. So although the peak was higher in HIV negatives, actually our HIV positive sustained response, which is very interesting. And we did look at the girls as well, although not as many of them, but what we were able to do is compare our girls to our data that is from Simon Dobson and Gina Ogilvie and others here, who looked at 2-dose, 3-dose in girls in a very, very similar format. So we had very comparable data in HIV negative girls and our HIV positive, and show some differences in the immune response to 18, 6, and 11, but not to 16. at 7 months post 3 doses, and then at 24 months, we see significantly better immune GMTs or geometric mean titers of antibody level in our HIV negatives compared to our HIV positives, which is certainly of concern. So we concluded that the vaccine was safe, well tolerated. We had high rates of seroconversion. The detectable viral load, so unsuppressed HIV virus, was correlated with a poor immune response, and important to do further follow-up. So Liz, I think I'll hand off to you here. Is that okay?
Yep, sounds good. So what I'm going to do now is just share a bit of the longer-term follow-up of the cohort and also some of the other sub-analyses that we did. So next slide, please. And so the first of these sub-analyses was actually going back to the baseline data and looking before vaccination, what were the oncogenic HPV types that were prevalent and persistent in this cohort. Next slide. So here you can see for each oncogenic HPV type, the percentage of participants who were experiencing a prevalent or persistent infection, and the most common ones are highlighted in red. And the most common ones were HPV 16, 45, and 52. And then I'll just bring your attention to the bottom of the slide where you can see that 45% of the women in this cohort had an oncogenic a prevalent oncogenic HPV infection, and a third of them were actually experiencing a persistent oncogenic HPV infection. So really high rates of prevalent and persistent infection prior to receiving the vaccine. And also, if you tally up the number of persistent infections that are caused by HPVs that are oncogenic but not contained in any available vaccine, 17% of these women had a persistent infection with those types. And so that suggests that we might really need to continue to focus on cervical cancer screening in this population despite vaccination. Next slide. So at baseline, we only had 7 cases of high-grade lesions, but we still wanted to look at those cases and see what types of HPV were co-occurring and could be cause of those lesions. And so we found that between 0 to 29% of the HSILs were due to HPV 16 or associated with HPV 16. None of them had a concurrent HPV 18 infection, and somewhere between 29 to 57% of the HSILs were due to non-valent HPV types, so the types that are added in the non-valent vaccine, or other high-risk types that are not in any available vaccines. So really the majority of the HSIL cases were not associated with HPV 16 or 18, again, suggesting that cervical cancer screening will remain important for these women. Next slide. Another sub-analysis that we did was to look at the impact that the spacing of the vaccine doses had, specifically on the peak antibody titer achieved in these women. And this work was published in Vaccine. Next slide. So the reason we were interested in this is because there's been a global movement towards reduced dose schedules and also altering the dose timing, but there was no data at all for women living with HIV. Specifically, the reduced dose schedules can pose significant cost savings. And I think Dr. Money also already mentioned that we ended up actually not having enough individuals who received only 1 or 2 doses to look at that, unfortunately. But in terms of wider dose spacing intervals, if there is a detrimental impact on the antibody titers achieved, then it suggests that we might need an additional booster dose of vaccine for those cases or to reinitiate the vaccine schedule. So we wanted to see if that was necessary. Next slide. So here you can see that we divided the cohort into women who had received all 3 doses within 7 months, within a span of 7 months to 1 year, or over the course of 1 to 2 years. And there were a few differences between these groups. So women with a wider dose spacing interval were more likely to have a lower CD4 count, to be not naive to HPV-11 and to have a longer time interval between when the last dose was administered and when they had a blood draw done for serology testing. Next slide. Here you can see the log peak HPV titers for those 3 different categories of dose spacing. And so right away you can see that there doesn't appear to be a huge difference between those 3 categories, potentially for HPV-18 in yellow. But we did a more in-depth analysis. So on the next slide. So here we looked at what were the predictors of the peak antibody response. And we found that here I'm only showing for HPV-16, but it was consistent between all 4 quadrivalent HPV types. So we saw that age was an important predictor, that HIV viral load being undetectable was an important predictor of the peak antibody response, as Dr. Money described, being naive to the HPV type, and also the time to blood draw after the last dose. And so what's really critical for this analysis is actually that the dose spacing category was never a significant predictor of the peak antibody titer. And so we have no evidence that restarting the vaccine schedule in these situations is needed, or providing a booster dose. And I also want to point out that all of these associations that we saw were expected. So if somebody's not naive to the HPV type, we expect them to have a higher antibody titer because their body's already been exposed to that pathogen. And then for the time to blood draw since the last dose, next slide. Here you can see what happens to the antibody titers over time, just to help explain this. So you have this peak in the antibody titer that usually occurs 1 month after the last dose, and then the titers sort of drop off and eventually plateau. So if the blood is taken further from the last dose, we expect the antibody titer to be a little bit lower. Next slide. Another analysis we did was to look at the 2-year efficacy of the vaccine. And this was published in Clinical Infectious Diseases at a time where there was no other efficacy data for women living with HIV. Next slide. And part of the rationale of doing this is that there's no known immune correlative protection for HPV. So while the immune titers give us some information, they don't give us the entire story. And it is important that we look at the efficacy to really inform how well the vaccine works. Next slide. So here you can see the endpoint rates. So we were looking at breakthrough persistent infection with those quadrivalent HPV types that these women have been vaccinated against, the rate of genital warts, and also the rate of SIN2+. In per protocol populations, naive to relevant type, and intention to treat populations. And so you can see we did have some cases of breakthrough infection and genital warts. We had no cases of SIN2+ at all in the cohort, but that was expected, being that it was only 2 years of follow-up and it takes a while for those endpoints to develop. Next slide. So here we focused in on the cases that occurred in the per protocol group. And again, there were low numbers, but we wanted to see, were there any stark indications of what could be contributing to these breakthrough infections? And so all of these women had received all 3 doses of vaccine. At the bottom, you can see the median CD4 count for the cases versus non-cases. And so again, the numbers are low, but the cases do appear to have a little bit lower of a baseline CD4 count and CD4 nadir. So this suggests that their present and historical immune compromise might be contributing. We're not sure yet. We'll need more cases to look at that. The HIV viral loads were, for the most part, well controlled. And then finally, in the far right column, we looked at the log peak HPV-18 antibody response. And the reason we looked at HPV-18 specifically for those QHPV cases is because all of the cases of breakthrough infection that we saw were HPV-18. It seemed a little bit statistically unlikely at the time, and so we're sort of interested in why that's the case. But Nonetheless, the peak antibody titers that we see for HPV 18 among the cases are actually a little bit higher, not different, but actually a little bit higher among the cases compared to non-cases. So it doesn't give us an easy explanation for why these women had these breakthrough infections. Next slide. We also did a comparison of a composite of all of these three endpoints to the literature from women without HIV. And in the two far right columns, you can see the rate ratios comparing our vaccinated women living with HIV to this cohort of women without HIV. And what you'll note is that the outcome rates were actually comparable in women living with HIV to the HIV negative placebo group, so women who had not received vaccine. What we would normally expect is that women living with HIV would have higher rates of these composite endpoints than women without HIV. So we are seeing these women have rates that have come down a bit, but not to the level of vaccinated women without HIV. Next slide. We also compared to the previous cohort that Dr. Money mentioned of women living with HIV in Canada. And here, drawing your attention to the persistent QHPV endpoint, we saw that there was an important reduction seen in the rate of persistent QHPV in vaccinated versus unvaccinated women living with HIV. Next slide. So overall, women living with HIV might be at higher risk for vaccine failure than women without HIV, but we do still think that there's an important benefit that we're seeing. And although the numbers are too low at that stage, the baseline CD4 count and CD4 nadir appear to be a bit lower among the cases, so they might be important predictors of vaccine failure. Next slide. Since then, we've actually looked at the efficacy beyond 2 years. So the most recent analysis, the median follow-up was just over 5 years. And what you'll see here is that the incidence rate has dropped dramatically for persistent QHPV as we've added more and more follow-up. So the case number has dropped because of the definition of persistent QHPV. So the definition is that these are HPVs that are present in at least 2 consecutive visits that are at least 6 months apart, or the HPV being present at the last available visit. And so it's known that this will overestimate the true rate a little bit, but it errs on the side of caution. So that's what the literature, what the community goes with. And interestingly, we're still seeing a dominance of HPV 18 in those cases of persistent QHPV. And still no cases of SIN2 plus in the per protocol population. There's been one case of SIN2 plus in the cohort, but it was an individual who had HPV 16 since the screening visit. Next slide, please. Okay, so now moving forward from here, we're going to tell you a little bit about a study that we have submitted to CIHR. So hopefully it'll be funded and upcoming. Next slide. So I love this slide because it really shows that, again, coming back to this goal of cervical cancer elimination, on the gray trajectory there, you can see that if we continue with our current vaccination and screening coverage, we are not going to reach the elimination goal. And so what it's going to take is intensive screening and vaccination in order to get there within the goal timeframe. What we're doing right now is not enough. Next slide. So how do we get there? We need scale up of screening and treatment. We need expanded vaccination programs. So right now there are school-based programs in many countries, but still no programs in many countries that could greatly benefit. So we need to add vaccine programs and also expand beyond these programs to target additional age groups and key populations. And we finally need new approaches and a deeper understanding of at-risk populations. And so that includes women living with HIV. Next slide. As Dr. Money told you in the beginning, HIV is a major global contributor to HPV incidence, and so we need an evidence-based plan. Evidence-based being the key bit there, because there's a serious lack of data in this arena. So we need an evidence-based plan to immunize women living with HIV prior to sexual debut across high-risk periods for acquisition and into adulthood. In the general population, there's been adoption of 2-dose vaccine schedules, and there's consideration of 1-dose vaccine schedules as well. There's a lot of ongoing studies looking at whether we can just use 1 dose, but it's still recommended that women living with HIV have 3 doses of vaccine. And again, this is just because there's a lack of data. There's literally no studies looking at reduced dosing in this population. And it's important because requiring additional doses for women living with HIV reduces the feasibility of vaccination for many women. And it also reduces the financial and logistic feasibility of vaccine programs in HIV endemic countries. Next slide. Even further to this, the quadrivalent vaccine has now been discontinued. There's only 1 publication on the non-valent vaccine in people living with HIV, and it's limited to only 100 individuals. So as a result of that, the National Advisory Committee on Immunization, our Canadian group, has named evaluation of non-valent vaccines in immunocompromised populations as a research priority. So we need data on the use of this non-valent vaccine in this population, as well as assessment of reduced dose schedules. Next slide. In response to this, we've proposed in the spring project grant competition a trial, a randomized clinical trial of non-valent vaccination in women living with HIV aged 18 to 50 across Canada who have not previously received any HPV vaccine. The participants will be randomized 1-to-1 to receive 3 doses of the vaccine at the routine schedule or at an expanded schedule. And I'll explain the reasoning behind that in a second. Next slide. So our primary research question is, are the peak geometric mean titers, so the peak antibody response to 2 doses, is it non-inferior to the peak antibody response to 3 doses? And that's measured at month 7, which is 1 month after the 2nd or 3rd dose. Secondary research questions are whether peak GMTs generated by the extended vaccine schedule are non-inferior to peak GMTs generated by the routine vaccine schedule, and whether the rate of antibody decay is greater among participants who receive the vaccine schedule in an extended versus routine format, following them up out to 24 months. Next slide. So I think this slide helps to sort of visualize what's actually going on here. So we have women get randomized to either the routine dosing schedule or the extended. And so those on the top in the routine dosing schedule receive all 3 doses of vaccine. And then their peak antibody titer is 1 month after that last dose. So it's at month 7. So we'll take a blood draw at that time and compare it to the month 7 blood draw from the other group, who at that same time point are experiencing their peak antibody response to 2 doses. We're also interested in comparing the peak antibody response to 3 doses in the top group, which will be at month 7, and in the bottom group, that will be at month 13. And then again, at month 24, comparing those 2 groups as well. So the expanded dosing interval lets us assess the peak antibody titers to 2 versus 3 doses at month 7, the primary outcome, while still offering the 3-dose standard of care to all women living with HIV in the study. Next slide. We're also going to look at the acceptability of HPV self-sampling as a mode of HPV testing among this population. And so the HPV DNA samples, we've proposed for those to be self-samples and to also provide a questionnaire at baseline and at the last visit regarding the acceptability and willingness to use that methodology of screening. And then we have exploratory aims surrounding safety and efficacy as well. Next slide. So we'll recruit 450 women living with HIV. That will be no small feat, but we have 11, longstanding engaged sites that have done this before. And we did a poll actually of the sites, and half of them were able to tell us how many women would be eligible at their site. So these are women who have not previously received any HPV vaccine. At only half of our sites, there are more than 900 eligible women. So it's actually really surprising how many unvaccinated women living with HIV there are in Canada. So hopefully, we get funded and we can also be providing vaccine to these women and they can benefit. It'll be a phase 2 trial because of the extended dosing interval, and all of the lab personnel will be blinded to the group allocation because they're assessing the primary endpoints, which are immunogenicity related. To be included, these individuals have to be living with HIV, have a uterine cervix, and be age 18 to 50. And then the exclusion criteria are quite standard for a vaccine trial. Next slide. This is how the visits play out, essentially getting their vaccine doses and the general safety follow-up that's associated with that, as well as blood draws for the immunogenicity endpoints, HPV DNA swabs to look at the efficacy endpoints, and the HPV self-sampling questionnaire at start and end of study to see how they feel about that screening methodology. Next slide. So the expected outcomes of this study, should it be funded, is that the project will provide globally relevant data to support evidence-based decision-making for HPV vaccination programs, and will ultimately help accelerate the global timeline towards cervical cancer elimination. There's a lot of novel information that we can provide through this design, so we're very hopeful. Next slide. Just to finish, we'd like to thank the study participants and clinical sites who contributed to all of that data that we presented over many, many years, and then also the sources of funding and support, which were CIHR, CTN, Merck, and the Department of OBGYN.